Hello, very good evening to the student who have joined. Can you please let me know if my voice is audible to you and if you can see the video? Thanks for confirming. Now I'm going to present, but just let me know that if the presentation is visible to you. Please confirm whether the PowerPoint presentation is visible to you or not. Okay, thanks for confirming. We will start at sharp 4 p.m. Please wait till now. We will start shortly, so kindly wait. Okay, so it's 4 p.m. and I think I will start with the introduction. So, okay, let me just make it a full screen. Okay, so very good afternoon to the students who have joined. My name is Mousumi Deb and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL course of Medicinal Chemistry and today it is our week 11 live session. So before starting the session, I would like to point out few things. So there will be the problem solving session every week and the sessions will be on Friday from 4 to 6 p.m. The sessions are live on Google Meet platform and this is the link for the every Friday class. The same link will be followed for every Friday class. So you can just write it down or copy it for somewhere and use the same link to join every day Friday class. So the important thing is if any network disruption happens from my side, do not leave the session immediately. I will try to rejoin as soon as possible. And if the students can post your doubts and queries through the chat box. Since I'm recording the session, okay, since I'm recording the session, I have muted your microphones or camera so that I can record the session seamlessly. But the chat box is always open and I'm monitoring the chat box every time. So if you have any doubt, any query, anything you want to tell me, you can use the chat box. Okay. After the session, if doubt persists, the students can drop an email to me. I will share my email ID to you, email ID with you, and also you can post your doubts or queries through the NPTEL forum also. Okay, the SWAM portal, you can use that also for asking your doubts. So as I have mentioned that I am recording the class and the class recording and presentation, which I am showing today, these things will be shared with you soon. Okay, so where to find these things? I have already mentioned it for a couple of times, but let me just show you one more time that you have to go to the SWAM portal. Okay, and in the SWAM portal, when there is a this particular announcement section, you will find there is a left panel, and in this left panel you will have one thing which is called the problem solving session January 2025. If you click on that, you will go to the problem solving session recording, and on clicking on that problem solving session recording, you will be directed to a page. Okay, and if you click here, you will be directed to a page which will contain the class recording of every week, and also along with the Google Drive link which contains all the lecture presentations. You can go to that particular drive and you can download that presentation and you can read from there. Okay, if you have anything, you have missed anything during the class. So this is the things. Just I thought to mention it one more time. Already I have mentioned it for a couple of times, but it's fine. Okay, so now today it is our week 11 live session. I have already mentioned it. So what we do, like every week, we have this kind of problem solving session and we solve MCQ type of questions in every class, right? So I will today also I will give you around of 20 MCQ questions, and all the questions will be based on the week 11 course content. This medicinal chemistry course is a 12-week course and already we are on our 11th week. So today we will solve the questions from the week 11 course content. Each question will have 4 different options. You have to let me know that among the 4 different options, which of the options will be correct. Okay, now after showing a question, I will wait for some time, and then I will check the chat box. I will check your answers, and after that, I will show you the correct answer, and we will discuss that particular topic in detail. And this way, it not only you will get time to practice your concepts and to understand the things, and also after this from the details discussion, you can also solve the subjective type of questions which may be asked to you. Okay, so this is the thing. And today, on one more thing, I would like to mention that today it is our week 11 live session. Next Friday will be our last live session, which is our week 12 live session, followed by a summary session which we scheduled on 13th of April, which is Sunday from 11 a.m. Okay, so do attend that session also. That will be our last session. And even if you have any doubts or any queries, you can let me know through email so that I can answer your queries in the last summary session. Okay. Now let me just go through the course content. So what are the course content of week 11? So week 11 majorly divided into the therapeutics based the antiviral therapeutics agents, anti-cancer therapeutic agents, cholinergics, and anti-ulcer agents. So basically this week course content is divided into three sections. One is based on the drugs which are antiviral, or we will discuss the little bit about the viruses also. Then will have our discussion on the anti-cancer agents. And then after that anti-ulcer agents, or with this are also known as the cholinergics. Okay, so let us go to the first question of today's session. So the problem number one, it says that which of the following statements is true regarding virus? Your options are: number A, viruses are not living organisms; number B, viruses grow and reproduce outside the host cells; number C, tuberculosis is a viral disease; and number D, viruses are giant compared to bacteria. Which of the following statements is true regarding the virus? Number A, viruses are not living organisms. Number B, viruses grow and reproduce outside the host cells. Number C, tuberculosis is a viral disease. And number D, viruses are giant compared to the bacteria. Which of the following statement will be true regarding virus? Let me know in the chat box, A, B, C or D, which of the following you think is the correct answer. After checking the answer, I will show you the correct answer and we will discuss it a little bit. Again, I will repeat the problem number one. It says which of the following statements is true regarding the virus? Your options are: number A, viruses are not living organisms; number B, viruses grow and reproduce outside the host cells; number C, tuberculosis is a viral disease; number D, viruses are giant compared to the bacteria. Which of the following statements is true regarding the virus? In the chat box, let me know. Since it is a practice session, I will not give you any negative marking for wrong answers. So please answer whatever you think is the correct one. Just go for it and let me know in the chat box. Okay. Let me just check the chat box, and after that I will discuss it. Okay, no answer. Any guesses? Which of the following may be the correct answer? Okay. Let me show you the answer and then let us discuss it a little bit. I hope my voice is audible, right? Even if you have any problem regarding the audio or anything presentation, please let me know in the chat box itself, okay? So the correct answer of problem number one, which says which of the following statements is true regarding the virus, is number A, which says that viruses are not living organisms. So this statement is true. Other three statements are false. Now let us discuss it a little bit. So two different terms we hear a lot, okay, regarding the diseases and all those things. So one is the virus and another is the bacteria. And there are the very some people they get sometime confused what is a virus and what is the bacteria. Okay, so there are some major differences between the virus and bacteria. And the first one is that viruses are not living organisms, okay, but bacteria are living organisms. Viruses only grow and reproduce inside the host cell. The infect. When found outside the living cell, viruses are dormant. Their life therefore requires the hijacking the biochemical activities of a living cell. But bacteria live in organisms that consist of a single cell that can generate energy, make its own food, move, reproduce, typically by binary fission. This allows bacteria to live in many places like soil, water, plants, human body, and serve many purposes. Viruses are submicroscopic. Bacteria are giant compared to the viruses. Okay, a viral infection is systematic. Viruses infect the host cell, infect a host cell, and then multiply by the thousands, leaving the host cell and infecting other cells in the body. But bacterial infection is usually confined to a part of body, described as a localized infection. Infections may be caused by a bacteria or by toxins, by the toxins produced. Okay, systematic diseases caused by viral infection include the influenza, measles, polio, AIDS, and COVID-19. Bacterial diseases include the pneumonia, tuberculosis, tetanus, and food poisoning. Okay, so these are the major differences between the viruses and bacteria. So let us just see our statements. So for the statement number 1 says viruses are not a living organism. This is true. The statement number B, it says viruses grow and reproduce outside the host cell. It is not true, because outside the host cell they cannot they cannot live. Okay, they need the host cell, they need the biochemical activities performed by the host cell to grow and reproduce. Okay, tuberculosis is a bacterial disease, it is not a viral disease, and viruses are not giant compared to bacteria. Bacteria actually are giant compared to the viruses. Okay, so the B, C, and D, these statements are false, but the statement number A is the true one. So what are the general characteristics of the viruses? So of the obligate intracellular, unable to self-replicate. Once inside the living cells, viruses induce the host cell to synthesize viral particles. The genome is either DNA or RNA, single or double-stranded. Viruses do not have a system to produce ATP. Viruses range in size from 25 to 270 nanometer. Viral tropism. Okay, the classification of viruses is based on the nucleic acid type, size, and shape of the virion, and presence and absence of the envelope. Viral structure. Virion is the entire viral particle. Capsid is a protein code that encloses the genetic material. Capsomer is the protein subunit that makes up the capsid. Nucleocapsid is composed of the capsid and genetic material. The envelope is the outer coating composed of the phospholipid bilayer, which is composed of the viral encoded glycoproteins and sometimes viral encoded matrix protein. The envelope is derived from the host cell's membrane. Some viruses use the plasma membrane, whereas others use the endoplasmic reticulum, Golgi, and nuclear membranes. Naked nucleocapsids are the viruses with no envelopes. Okay, so these are the general characteristics of the viruses. Okay, so is it fine, or there is any doubt from problem number 1? Okay, I think I have got one answer for previous problem. Okay, it's fine. Now I will go to the next problem. If it is clear, I will go to the next problem. Okay. Yeah. So problem number 2 says, which of the following statements is false regarding the characteristics of the good protein target for a antiviral drug? Your options are: number A, it should be important to the life cycle of the virus. Number B, it should bear little resemblance to the human proteins. Number C, it should be important in the late stages of the virus life cycle. Number D, it should be common to different types of virus. Which of the following statements is false regarding the characteristics of the good protein target for antiviral drugs? Again, I will repeat the options. Number A, it says it should be important to the life cycle of the virus. Number B, it should bear a little resemblance to the human protein. Number C, it should be important in the late stage of the virus life cycle. Number D, it should be common to different types of viruses. Which of the following statement is false regarding the characteristic of a good protein target for antiviral drugs? Let me know in the chat box A, B, C, or D which of the following you think is will be the correct option. I will wait for a minute, then I will check the chat box. Okay. So which of the following statement is false regarding the characteristic of a good protein target for antiviral drugs? Number A, it should be important to the life cycle of the virus. Number B, it should bear little resemblance to the human proteins. Number C, it should be important in the late stages of the virus life cycle. Number D, it should be common to different types of virus. Let me check the chat box. No answer. Okay. So let me show you which of the following will be the correct answer. So the correct answer for problem number 2 will be number C. It should be important in the late stages of the virus life cycle. This statement is false regarding the characteristics of the good protein target for antiviral drugs. Okay. So antiviral drugs, it can be two type majorly. One is the targeting the viral function, and second is the targeting the cellular function that virus needs. Okay. So the targeting the viral function can be of four different types. One is the nucleic acid polymerase, then the protease, integrase, and neuraminidase. So these are the targets which can be targeted by this antiviral agents. So nucleic acid polymerase can be three types: DNA dependent DNA polymerase, RNA dependent RNA polymerase, and DNA dependent RNA polymerase. Okay, so these are the very good antiviral drug targets. So what are the major characteristics of the antiviral drug? First, it should be able to enter the cells infected with virus, then interfere with the viral nucleic acid synthesis or regulation. Some drugs interfere with the ability of the virus to bind to cells. Some drugs stimulate the body's immune system. Best responses to antiviral drugs are in the patients with the competent immune systems. A healthy immune system works synergistically with the drug to eliminate or the suppress the viral activity. Okay, so these are the key characteristics of the antiviral drugs. So problem C was the correct answer for, option C was the correct answer for problem number 2. Okay, I have already mentioned it that there is no negative marking for answering during the class. So please try to answer the question. Even if your answer is wrong, it does not matter, but please try to answer. Okay, whatever you think is the correct answer, let me know in the chat box. Now I will go to the next problem. So problem number 3 says identify the correct order of the viral infection in the host cell. Okay, you have to identify the correct order of the viral infection in the host cell. Your options are number A, attachment, followed by uncoating, followed by penetration, followed by release, assembly, and biosynthesis. Number B, attachment, then penetration, uncoating, biosynthesis, release, and assembly. Number C, attachment, biosynthesis, penetration, uncoating, assembly, and release. And number D, attachment, penetration, uncoating, biosynthesis, assembly, and release. Which of the following you think is the correct order for the viral infection in a host cell? Number A, attachment, uncoating, penetration, release, assembly, biosynthesis. Number B, attachment, penetration, uncoating, biosynthesis, release, and assembly. Number C, attachment, biosynthesis, penetration, uncoating, assembly, and release. Number D, attachment, penetration, uncoating, biosynthesis, assembly, and release. Which of the following will be the correct order for the viral infection in the host cell? Let me know in the chat box A, B, C, or D which of the following you think is the correct answer. Identify the correct order of the viral infection in the host cell. Let me check the chat box. Okay, I have got one answer which is saying it is number B. Okay, let me check if number B is the correct answer or not. So the correct answer for problem number 3 will be, it is number D. Attachment followed by penetration, uncoating, biosynthesis, assembly and release. Okay, so let me just show you. So what are the steps of the viral infection? So the most viruses follows the similar series of steps in the viral reproductive cycle. Okay, so these are the attachment, penetration, uncoating, replication, or replication or biosynthesis, assembly, and then release. Once inside the host cell, the viral DNA or RNA is released and can begin the to interact with the internal cell machinery. Viral DNA can direct the transcription of the viral mRNA, which is then translated into the viral proteins. Viral RNA can be used as a template to make more viral mRNA and proteins. RNA retroviruses use the reverse transcription to make the viral DNA, which is then transcribed or translated. Regardless of the mechanism, the objective is the same: more variants. New variants are released when damage to the host cell causes lysis, or when the host cell undergoes apoptosis. Then some viruses are released through the budding, which releases one variant at a time and spurs the host cell. Okay, so these are the steps of the viral infection. So in short, first is the attachment or the adsorption. Okay, once when the virion is attached to the host cell, then is the penetration, which happens by fusion. Then this virion is engulfed by the host cell, as you can see, and this is the glycoprotein which takes up the virion, and then the virion is enters the host cell, followed by the uncoating. Okay, this uncoating releases the viral materials in the host cell. Then is the biosynthesis or replication or the translation or transcription, whatever it is, the replications of the viral virion particles. Then the assembly, okay, assembly and synthesis assembly of the nucleocapsids, and followed by the release. Okay, and the release happens by the budding. So budding is the opposite to the penetration, when this particular then viral proteins incorporated into the cell membrane, and then the release. So this is the life cycle of the DNA virus, such as the herpes simplex. This is the example, and these are the different steps. Okay, so number D was the correct answer for problem number 3. Now let me go to the next problem. Problem number 4 says, which of the following enzymes is responsible for incorporating viral DNA with the host DNA? Your options are: number A, reverse transcriptase; number B, protease; number C, integrase; number D, DNA polymerase. Which of the following enzymes is responsible for incorporating viral DNA with the host DNA? Reverse transcriptase, protease, integrase, DNA polymerase. Which of the following enzymes is responsible for incorporating viral DNA with host DNA? Let me know: A, B, C or D. Which of the following will be the correct answer? Which of the following enzymes is responsible for incorporating viral DNA with host DNA? Again, I will wait for a minute, then I will check the chat box and we will discuss this particular question. Okay? Let me check the chat box. Okay, I got one answer which is saying it is the number A. Let me check if number A is the correct answer or not. So for the problem number 4, the correct answer will be, it is number C, integrase. Integrase is the enzyme which is responsible for incorporating the viral DNA with the host DNA. So this is a structure of the HIV particle. Okay. So as I have already mentioned, the outer layer is the envelope, then followed by the genetic materials. We have this the different enzymes like reverse transcriptase, integrase, and the capsid, which actually covers the genetic materials of the cell. So this is the life cycle which I have mentioned in the last slide. So first is the absorption or the attachment of the virions to the host cell. Okay, and the CD4 is the where it binds the glycoprotein. Then after the penetration, what happens? The virus they have their RNA, and this RNA is converted to the RNA DNA hybrid by the reverse transcriptase, and which converts to the single strand DNA and followed by the double strand DNA by the reverse transcriptase enzyme. Now this viral DNA, which is synthesized inside the host cell, is incorporated to the host DNA. Okay, and the enzyme which actually does this work is the integrase enzyme. As you can see, this black one is the host DNA and the sky blue one is the viral DNA. So what does this integrase is doing? It is incorporating the viral DNA to the host DNA. Okay, and then it follows the RNA, and this RNA will synthesize the viral proteins, which will give rise to the new virions, and which will be released from the host cell by the budding technique. Okay, which is this one. So remember, there are few enzymes which are very important in the virus life cycle. Integrase is one of them. Protease is also there. Reverse transcriptase. All these enzymes are very important. But the in particular work that integrase does is it actually fuses the viral DNA with the host DNA so that it can generate the viral proteins via RNAs. Okay, so this is the work or this is the mechanism of action and how integrase works and how the viral life cycle goes on. Okay. Now I will go to the next problem. So problem number 5 says, which of the following drugs is used for the treatment of CMV retinosis? Your options are number A, desciclovir, number B, valacyclovir, number C, trifluridine, number D, foscarnet. Which of the following drugs is used for the treatment of CMV retinosis? Desciclovir, valacyclovir, trifluridine, or foscarnet? Which of the following drugs is used for the treatment of CMV retinosis? Let me know in the chat box, A, B, C, or D, which of the following you think is the correct answer for problem number 5. Which of the following drugs is used for the treatment of CMV retinosis? Desciclovir, valacyclovir, trifluridine, or foscarnet? I will wait for a minute and then I will check the chat box. So, which of the following drugs is used for the CMV retinesis? Let me check the chat box now. Another answer I have got, which is saying it is number A. Okay, let me check if number A is the correct answer or not. So the correct answer will be number D, foscarnet. So foscarnet is the drug which is used for the treatment of the CMV retinesis. So clinical aspects of the viral DNA polymerase inhibitor. So I have already mentioned that DNA polymerase enzyme is a very good antiviral drug target, and there are several DNA polymerase inhibitors which are used as an antiviral therapeutic. Okay, so these are the several examples. Acyclovir, which is represented a revolution in the treatment of the herpes infections, being the first relatively safe non-toxic drug to be used systematically. It is used for the treatment of the infections due to the herpes simplex 1 and 2, that is the herpes simplex encephalitis and the genital herpes, as well as the varicella zoster virus, which is the VZV, that is the chickenpox and shingles. Unfortunately, strains of the herpes are appearing which are resistant to acyclovir. So this can also rise as a result of the mutations either in the viral thymidine kinase enzyme, such it no longer phosphorylates acyclovir, or of the viral DNA polymerases, such it no longer recognizes the activated drug. Acyclovir is not effective against all types of herpes virus. There are 8 herpes viruses that are divided into 3 subfamilies. Acyclovir is effective against the alpha subfamily, but not for the beta subfamily, because later produces a different thymidine kinase that fails to phosphorylate the drug. Now, valacyclovir is a valine prodrug for the acyclovir and is particularly useful in the treatment of the VZV infections. When this prodrug is given orally, blood levels of the acyclovir is obtained, which is equivalent to those obtained by intravenous administration. Decyclovir is another prodrug for acyclovir, but some more toxic and thus limiting the potential. Ganciclovir is phosphorylated by the thymidine kinases produced by the both alpha beta subfamilies of herpes virus and can be used against both viruses. Unfortunately, the drug is not safe as acyclovir because it can be incorporated into the cellular DNA. Nevertheless, it can be used in the treatment of the CMV infections, okay, which is the cytomegalovirus. This is a virus which causes eye infection, can lead to blindness. Acyclovir is not effective in the this infection because CMV does not encode a viral thymidine kinase. Ganciclovir, however, can be converted to its monophosphate by kinases other than the thymidine kinase. As ganciclovir has a low oral bioavailability, the valine prodrug vanciclovir has been introduced for the treatment of CMV infections. Penciclovir is essentially same spectrum of the activity as acyclovir, but has better potency, faster onset, and longer duration of action. It is used topically in the treatment of the cold sores in HSV1 and intravenously in that for the treatment of HSV in the immunocompromised patients like acyclovir. Penciclovir is also has a poor bioavailability and poorly absorbed from the gut because of the polarity. Therefore, famciclovir is used as a prodrug for better absorption. Cidovovir is a broad spectrum antiviral agent which shows the selectivity for the viral DNA polymerase and used to treatment the retinal inflammation caused by the CMV. Unfortunately, the drug is extremely polar and has a poor bioavailability. It is also toxic for kidneys but can be reduced by co-administrating with the probenecid. Idoxuridine can be used for the topical treatment of the herpes scateresis, but trifluridine is a drug of choice for this disease because it is effective at the lower dose frequencies. And vidravin is an early antiviral drug, but acyclovir is now generally preferred. Foscarnet is used for the treatment of CMV retinesis, which is approximately equal activity of the ganciclovir. It is also be used in the can be used in the immunocompromised patients for the treatment of HSV and VZV strains which are prone to resistant to acyclovir. It does not undergo any metabolic activation. So these are all the clinical aspects, examples, and the therapeutic areas for the different DNA polymerase inhibitors which are used as antiviral agents. Okay, you don't need to memorize all these things. I have just given it for your knowledge, but try to remember which drug is used for which particular infection, the clinical aspects. Okay, so this thing you should be remembering. So this is the structure of the phoscarnet, which I have already mentioned. Okay, this is used for the CMV retinosis, and also it can be used in the immunocompromised patients for the treatment of HSV and VZV strains which are proven resistance to the acyclovir. So now I will go to the next problem. Problem number 6 says which of the following statements is false regarding NRTIs. Okay, your options are: number A, they have good oral bioavailability; number B, they act against both HIV 1 and 2; number C, they are less toxic than NNRTIs; and number D, they are excreted through the kidneys. Which of the following statements is false regarding NRTIs? Number A, they have good oral bioavailability. Number B, they act against both HIV 1 and 2. Number C, they are less toxic than NNRTIs. Number D, they are excreted through the kidneys. I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct option. Which of the following statements is false regarding the NRTIs? Let me just check the chat box. Okay. Okay, I think there is no answer for this question. It's fine. Let me just show you the correct answer and let us discuss it a little bit. So the statement which is false regarding the NRTIs is number C. They are less toxic than NNRTIs. Okay, so this statement is false. So what are the clinical aspects of this RTI or the reverse transcriptase inhibitors? Okay, RTI means reverse transcriptase inhibitors, which are mainly of two types. One is the NRTIs, which is the nucleoside reverse transcriptase inhibitor, and one is the non-nucleotide reverse transcriptase inhibitors or NNRTIs. Okay, so non-nucleoside reverse transcriptase inhibitors or NRTIs are currently used as a part of the combination therapy combating the HIV. Generally, NRTIs have good oral bioavailability, are bound to minimally to plasma proteins and are excreted through the kidneys. They also act against both HIV 1 and 2. However, they are often associated with the toxic side effects. Zerobodine was the first anti-HIV drug to reach the market, but can cause severe side effects such such as the anemia. Deronosine was the second anti-HIV drug approved for the use and reached US market in 1988. However, there is a risk of the toxicity in of the pancreas. Abacavir was approved in 1998, but been used successfully in the children in combination with the protease inhibitors nelfinavir and saquinavir. However, life-threatening hypersensitivity reactions have been reported in some patients. Tenofovir disoproxil was approved for HIV-1 treatment in 2001. It remains infected cell longer than many other antiretroviral drugs, allowing for once daily dosing, but can have toxic effects on the kidneys. It can be used alongside the emtricitabine, which is relatively free of the toxic side effect. Other NRTIs used against the HIV include the lamivudine and stavudine. Lamivudine is less toxic than the zidovudine, has also been approved for the treatment of hepatitis B. Zalcitabine is an NRTI which acts against the hepatitis B, but long-term toxicity means it is unacceptable for the treatment for the chronic viral diseases which are not life-threatening. Adefovir was approved by the US Food and Drugs Administration, or FDA, in 2002 for the treatment of the chronic hepatitis. It is also active on the viruses such as the CMV and herpes. NNRTIs, or non-nucleoside reverse transcriptase inhibitors, compared to NRTIs, these show higher selectivity for HIV-1 reverse transcriptase over the host DNA polymerase. As a result, these are less toxic and have fewer side effects. Unfortunately, lapid resistance emerges if an NRTI is used of its own. Thus, it does not occur if an NNRTI is combined with an NRTI from the start of the treatment. NNRTIs are restricted to the HIV-1 activity and generally metabolized by the liver. They can also interact with the other drugs and bind more strongly to plasma protein. Nevirapine, delaverdine, etravirdine, these are the NNRTIs currently approved by the FDA for the treatment of the HIV. Okay. So these are the different chemical aspects for the NRTIs and NNRTIs. You just have to remember what are the characteristics of the both and also few examples. Okay, so like the question can be asked, you can be given a particular name of a drug and you can be asked to identify if it is a NRTI or if it is a NNRTI. Okay, and all the mechanism of action. So these are the important things which you need to remember from these topics. Okay. Now I will go to the next problem. Problem number 7 says the HIV protease enzyme is symmetrical, whereas the mammalian proteases are not. What significance might this have? Again, I will repeat, the HIV protease enzyme is symmetrical, whereas the mammalian proteases are not. What significance might this have? Your options are: number A, inhibitors of HIV protease enzyme must be symmetrical. In number B, it may be possible to design inhibitors that prove selective for HIV protease over mammalian protease. Number C, only symmetrical substrate can be cleaved by HIV protease. Number D, there is no significance. Again, I will repeat. HIV protease enzyme is symmetrical, whereas mammalian proteases are not. What significance might this have? Number A, inhibitors of HIV protease enzyme must also be symmetrical. Number B, it may be possible to design inhibitors that prove selective for the HIV protease over mammalian protease. Number C, only symmetrical substrates are cleaved by HIV protease. Number D, there is no significance. I will wait for 2 minutes, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 7. Try, even if your answer is wrong, it's fine, but try to answer the question. Let me check the chat box now. Okay, there is no answer. Okay, fine. Let me just show you the correct answer and let us discuss it a little bit. So, the significance which this particular phenomena might give rise to is the correct answer will be number B. It may be possible to design inhibitors that prove selective for the HIV protease over the mammalian protease. So the HIV protease enzyme. So HIV protease enzyme is a symmetrical dimer. Let me just run the list point. Okay, so the HIV protease enzyme is a symmetrical dimer made up of two identical protein units, each consisting of 99 amino acids. The active site is at the interface between the protein units and also symmetrical with the twofold rotational symmetry or cytosymmetry. The enzyme has a broad substrate specificity and can cleave a variety of peptide bonds in a viral polypeptide, but crucially it can cleave the bonds between the proline residue and an aromatic residue like phenylalanine or tyrosine. Moreover, the symmetrical structure of the viral enzyme is and the active site is not present in the mammalian proteases, again suggesting the possibility of drug selectivity. So whenever there is an enzyme in the virus or any bacteria which we want to target, and a similar kind of enzyme like protease is the enzyme which we also have in the mammalian normal healthy cells and all the viral cells. Okay, so for having this kind of antiviral drugs or the drugs which will target this particular viral enzyme, it is very necessary that particular drug or the therapeutic agent targets that enzyme selectively. It does not harm any the mammalian proteases or the normal cells. Same goes to for the like the anti-cancer drug. They have to be selective for the healthy cell and cancer cell. The thing is same also here. Okay, so the structure, this particular symmetric dimer structure is for the HIV protease is different from the mammalian protease, and this structural difference can be targeted when we are going to design an antiviral agent as a HIV protease inhibitors. And protease inhibitors are very crucial antiviral therapeutics, a crucial class of antiviral therapeutics, especially in the case of the HIV, because there are several drugs which are marketed as a protease inhibitors, which shows very good anti-HIV activity. Okay, so number B is the correct answer. Now I will go to the next problem. Problem number 8 says on which receptor T cell surface HIV binds. Your options are number A CD4, number B CD6, number C CD7, and number D CD2. On which receptor of T cell surface the HIV binds? CD4, CD6, CD7, or CD2? I think you can answer this question easily because I have already mentioned it one time in our first or second question itself, if I remember correctly. So I will give you some time. I will give you a minute and then I will check the chat box. On which receptor of T cell surface the HIV binds? CD4, CD6, CD7, and CD2. Let me know in the chat box. Let me check the chat box. Okay, I have got one answer which is saying it is number C. Let me check if number C is the correct answer or not. So the receptor of the T cell surface where the HIV binds is the number A, CD4 receptor. Okay. So this is the life cycle of a HIV virus. So it is same which I have already mentioned in the second question itself. So first is the attachment or the binding or the absorption. So is the on the surface of the T cell, the HIV binds to a CD4 receptor and one or two co-receptors like CXCR4 and CCR5. So this is a very important thing which you need to remember. Okay, on the surface of T cell, the HIV binds to the CD4 receptor and one or two co-receptors like CXCRV4 and CCR5. Then is the fusion. The virus fuses with the host cell. So this is the HIV virus. This is the structure I have already mentioned. We have this GP41 and GP120. Then we have this here, as you can see, there is a CD4 or CCR5. Okay, and it is absorbing. This receptors are absorbing the virus virion. This we have the enzymes like integrase, reverse transcriptase, and the capsid is covering the genetic material, and we also have the virus envelope. So then fusion, the virus fuses with the host cell membrane and releases the genetic material, the RNAs to the cell. Here the RNAs are released to the cell, and this is the RNA, and there is a reverse transcriptase synthesize the viral DNA. The reverse transcription is the third step where the single stranded HIV RNA is converted to the double stranded HIV DNA by the reverse transcriptase enzyme. Fourth step is the integration. After the HIV DNA enters the cell's nucleus, the enzyme integrase cuts the cell's DNA, inserts the HIV DNA to the cell's DNA. Then is the transcription and the translation. The enzyme RNA polymerase makes RNA copies of DNA, and HIV RNA is either inserted to the new virus particle or it is to be translated to synthesize the HIV proteins. And then number 6 is the assembly. The long protein chains are cut into the individual proteins by the enzyme HIV protease, and new viruses are assembled with these proteins and the HIV RNA. And then 7 is the release. The new virus particle is released from the host cell, taking all the part of the cell's membrane are capable of infecting the new cells. So this is the complete life cycle of the HIV virus. Okay, how it works. So the steps already I have mentioned earlier. Also, I will again repeat. First is the binding. Second is the fusion. Third is the this reverse transcription. Then we have this transcription and translation. Then we have the integration, then reverse, then transcription and translation. Sixth is the assembly, and seventh is the release. Okay, so CD4 and CXCR4 and CCR5 are the receptors and co-receptors respectively for the where this HIV binds on the T cell. Okay, now I will go to the next problem. Problem number 9 says, which of the following statements is false regarding the adamantins? Your options are: number A, they are known as the ion channel disruptors. Number B, they inhibit the replication of the influenza A virus by blocking the matrix protein channel. Number C, with the change in the concentration, their mechanism of action changes. Number D, the virus cannot mutate in the presence of amantadine. Which of the following statements is false regarding the adamantins? Number A, they are known as ion channel disruptors. Number B, they inhibit the replication of the influenza A virus by blocking the matrix protein channel. Number C, with the change in the concentration, their mechanism of action changes. And number D, the virus cannot mutate in the presence of the amantadine. I will wait for a minute or two, then I will check the chat box. After that, I will discuss these things. Okay. Read all the options very carefully, because in the true false type of question the students sometimes make mistake by not reading the options very carefully. Which of the following statements is false regarding adamantins? Let me check the chat box now. Okay, I have got one option which is saying it is number C. Let me check if number C is the correct answer or not. So, the statement which is false regarding the adamantins will be this number D. The virus cannot mutate in the presence of amantadine. So ion channel disruptors or adamantins. The adamantins, these are the figures. Amantadine is, this is the structure of amantadine and this is the structure of rimantadine. So adamantins were discovered by the random screening and are earliest antiviral drugs used clinically against flu, decreasing the incidence of the disease by 50 to 70%. Amantadine and rimantadine are related adamantins with similar mechanisms of action and can inhibit viral infection in two ways. At a low concentration, less than 1 microgram per ml, they inhibit the replication of the influenza A viruses by blocking the viral ion channel protein called matrix M2 protein. At higher concentration, greater than 50 microgram per ml, the basic nature of the compound becomes important and they buffer the pH of endosomes to prevent the acidic environment needed for the HA to fuse with the viral membrane with the endosome. These mechanisms inhibit the penetration and uncoating of the virus. Unfortunately, the virus can mutate in the presence of the amantadine to form the resistant variants. The amantadine binds to the specific region of M2 ion channel. The resistant variants have mutations which alter the width of the channel. Research carried out find analogs of might still bind to this mutant prove unsuccessful. Work has been carried out on an attempt to find an analog which might affect the ion channel and the pH level at a comparable concentration. This has focused on the secondary tertiary amine with increased basicity as well as alteration of the structure to reduce the activity of the ion channel. The rationale of the resistant flu variants are less likely to be produced if the drug acts on the two different targets at the same time. Rimantadine was approved in 1993 as a less toxic alternative to amantadine for the treatment of influenza A. Unfortunately, neither agent is effective against influenza B as the virus does not contain the matrix M2 protein. Side effects are also is a problem, possibly owing to the effects of the host cell ion channels. Okay. So, statement number D, which says that virus cannot mutate in the presence of amantadine, is the not true. It is the false. Okay. Now I will go to the next problem. So problem number 10 says identify the statement which is incorrect. Your options are number A, DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, a DNA synthesis checkpoint is present. Number C, cancer can result when one or more cell cycle checkpoints malfunction. Number D, G2 phase occurs between the S and M phase. Identify the statement which is incorrect. DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, the DNA synthesis checkpoint is present. Number C, cancer can result when one or more cell cycle checkpoints malfunction. And number D, the G2 phase occurs between S and M phase. So now, till now, we were discussing about the antiviral therapeutics or antiviral agents. Now we are going to the anti-cancer agents. Okay, so for the couple of for the rest of the time, we are going to discuss on the anti-cancer agents and the anti-ulcer drugs a little bit. So let me know A, B, C, or D. Which of the following you think is the correct answer for problem number 10? I will wait for a minute, then I will show you the correct answer and we will discuss it a little bit. Let me check the chat box now. So I have got one answer which is saying it is number A. Let me check if number A is the correct answer or not. So the statement which is incorrect will be, very good, it is number A, which says that DNA synthesis checkpoint exists in the growth phase of the cell cycle. So this is the cell cycle and the checkpoints. The following diagram describes what the different checkpoints in cell cycle are responsible for. So the first one is the G1, which is the cell growth cell growth phase, and the first checkpoint is the cell growth checkpoint, which has occurs toward the end of the growth phase 1. Okay, and in the growth phase 1 also we have a resting phase, resting period G0. So what happens? So what does this cell growth checkpoint do? That it occurs toward the end of the growth phase 1 and checks whether the cell is big enough and has made proper proteins for the synthesis phase. If not, the cell goes to the resting period G0 until it is ready to divide. Then we have the DNA synthesis checkpoint, and this DNA synthesis checkpoint occurs during the synthesis phase S. Okay, it checks whether the DNA has been replicated correctly, and if so, the cell continues to mitosis, onto mitosis or M. Then we have this G2 phase, which occurs between S and M phase, and followed by a mitosis checkpoint in the M phase. It occurs during the mitosis phase and checks whether the mitosis is complete. If so, the cell divides and the cell cycle repeats. So these are the cells. So major the phases are the G1, S, G2, and M. Okay. There are four major phases, and one is the G0 is the resting phase, and the three checkpoints, cell growth checkpoint occurs at the end of the growth phase 1, synthesis checkpoint occurs in the S phase, and mitosis checkpoint occurs in the M phase. Okay, cancer can result when the one or more of these checkpoints malfunction. Basically, a stalled DNA replication fork will activate the S-phase checkpoints where it is ensured the proper replications of the genetic material. If this checkpoint is somehow missed, the another checkpoint at G2 is re-ensure proper replication, devoid of any DNA damage prior to initiation of the mitosis cycle. Okay, so here all other checkpoints as I have mentioned. So majorly there are three checkpoints. One is the damaged DNA checkpoint, which is the first cell growth checkpoint. Then unreplicated or damaged DNA is checked here. Then if the unreplicated damaged DNA is present, it is checked here also, and if chromosome is misalignment is checked here in the M phase. Okay, so these are the cell growth checkpoints and cell cycle phases. Now I will go to the next problem. Problem number 11 says, which cyclin is involved in balancing the cell growth and division? Your options are number A cyclin A, number B cyclin B, number C cyclin D, and number D cyclin E. Which cyclin is involved in balancing the cell growth and division? Cyclin A, B, D, and or E? Which cyclin is involved in balancing the cell growth and division? Cyclin A, B, D, or cyclin E? Let me know in the chat box which will be the correct answer. Which cyclin is involved in balancing the cell growth and division? I will wait for a minute, then I will check the chat box. Which cyclin is involved in balancing the cell growth and division? Okay, let me check the chat box because we have many questions left till now. Okay, no answer is fine. Let me check the which of the following will be the correct answer. So, the cyclin which is involved in balancing the cell growth and division is number C, cyclin D. Okay, cyclin D is involved in balancing the cell growth and division. So, control of the cell cycle by cyclins and cyclin dependent kinases. So, basically, the control of the cell cycle involves a variety of protein called cyclins and the enzyme called the cyclin-dependent kinases or CDKs. There are at least 15 types of cyclin and 9 types of CDK. Each has a role to play at different stages of the cell cycle. Examples are shown in this figure. So, in case of the growth phase is mainly the G1 phase. We have the cyclins 4 and 6. Okay, and cyclin the CDK cyclin CDK or cyclin dependent kinase 4 or 6 and cyclins D1, D1 to 3. And then at the end of the G1 after this checkpoint, we have this CDK2 kinase and cyclin E protein. In the S phase, majorly there is a CDK2 kinase and we have the cyclin A. In the CDK2 and cyclin A are also present in G2 and CDK1 and cyclin B are in the M phase. Okay. Binding of a cyclin which is associated with the kinase activities and the enzymes serves to move the cell from one phase of the cell cycle to another. For example, when a cell is in the G1 phase, the decision has to be made whether to move into the S phase or to start copying the DNA. This decision is taken independently on the balance of the stimulatory versus the inhibitory signals being received through the signal transduction. If this balance is toward the cell growth and the division, there is an increase of the cyclin D. This indicates that this binds to the CDK4 and CDK6. The resulting complex is phosphorylated powerful growth inhibitory molecule known as the PRB, which normally binds and inactivates the transcription factor. Phosphorylation altered the PRB such as can no longer bind to the transcription factor, and the latter is free to bind specific regions of the DNA. This results in the transcription of the specific genes, which leads to the production of the proteins capable of moving the cell towards the S phase. For example, cyclin E and thymidine kinase. Once cyclin E has been produced, it combines with the CDK2 and this complex is responsible for the progression from the G1 phase to S phase. Other activated cyclin CDK complexes are more important in the different phases of the cell cycle. For example, cyclin A CDK2 complex is required for the progression through the S phase and the cyclin B CDK1 complex is necessary for mitosis. Okay. So, the major cyclin which is involved in the cell growth and division is the cyclin D. Now I will go to the next problem. Problem number 12 says palbociclib is the inhibitor of dash kinase. Your options are number A JAK, JK, number B BCR-ABL, number C CDK4/6, and number D MET. Palbociclib is the inhibitor of which kinase enzyme? JAK kinase, BCR-ABL kinase, CDK4/6 kinase, or MET kinase? Let me know A, B, C, or D. Which of the following you think is the correct answer? Palbociclib is the inhibitor of which kinase? Palbociclib is an inhibitor of which kinase? JAK kinase, BCR-ABL kinase, CDK4/6 or MET kinase? I will wait for a minute, then I will check the chat box. Let me check the chat box. Okay, let me show you the correct answer and let us discuss it a little bit. So palbociclib is an inhibitor of CDK4 and 6 kinase. So cell cycle inhibition by CDK4/6 inhibitors. So I have already mentioned that cyclin D and CDK4 this complex are responsible in the growth phase. Okay. So basically in the G1, the cell accumulates resources to need to duplicate the genome, and the G0 is the quiescent state where the cell rests. Then we have the S phase where the actual duplication of the genome occurs, then followed by the proper DNA replication checked in the final preparation for the mitosis in the G2 phase, and the chromosomes are separated and actual cell division occurs is the M phase. So side CDK4/6 inhibitors, what does they do? They basically acts on this G1 phase. Okay, so palbociclib or Ibrance is an inhibitor of the CDK4 and 6. This is the structure. Okay, so this is a type 1 kinase inhibitor, and the primary target is CDK4/6, and this binds to the ATP pocket of an active conformation through 2 hydrogen bonding with the kinase hinge through the valine 101 and one with the aspartate 163 of the DFG motif. Its cyclopentyl group occurs in the ribosome pocket. Treatment of the hormone responsive or HR positive, HER negative or advanced or metastasis breast cancer in the postmenopausal woman. Side effects is the neutropenia and leukopenia. Okay, so this is the clinical aspects of the palbociclib, which is a kinase inhibitor, and kinase inhibitor actually a very big class of the enzymes and therapeutic agents as which acts as a majorly in the anti-cancer drug discovery, and we will discuss it in a little bit how this kinase inhibitors work and all these things. Okay, so the correct answer was number C. One thing I need to just correct here is it will be okay. Now I will go to the next problem. Problem number 13 says, what is the general term used to indicate the growth of the new blood vessels? Your options are number A, biosynthesis, number B, apoptosis, number C, angiogenesis, number D, metastasis. What is the general term used to indicate the growth of the new blood vessels? Biosynthesis, apoptosis, angiogenesis, or metastasis? Let me wait for a minute, then I will show you the answer and we will discuss it. What is the general term used to indicate the growth of new blood vessels? Let me check the chat box. I have one answer which is saying it is number D. Okay, let me check if number D is the correct answer or not. So the term which indicates the growth of the new blood vessels is number C, angiogenesis. So angiogenesis is basically, just let me show you. So it's defined as the formation of the new capillaries from the pre-existing vasculature by the migration and the proliferation of the endothelial cells. Okay, so as the tumor grows, its cancerous cells require the steady supply of amino acid, nucleic acid, bases, carbohydrate, the oxygen and growth factors if they are to continue multiplying. This means that the tumor has to have a good blood supply. As the tumor grows in size, however, its cells become increasingly remote from the blood supply and become starved of these resources. Oxygen levels also fall, resulting in the state of the hypoxia. This is particularly true for the cells from the center of a tumor. As a result, this hypoxia-inducible factors such as HIF1 start to build up within the tumor cell, and these factors serve to upregulate the genes to promote the survival of the oxygen-starved environment. For example, growth factors are released from the cell, and such as the vascular endothelial growth factor VEGF and fibroblast growth factor FGF2, which interact with the receptors on the endothelial cells of the nearby blood vessels and stimulate these cells to divide, leading to the branching and the extension of the existing capillaries, a process known as the angiogenesis. Okay, so this is the how angiogenesis happens. So this is the tumor cell. So when the tumor cell grows in size, what happens? This basically this VEGF and FGF2, these are triggered, these factors, and we have this endothelial cell and blood vessels. So angiotensin and thrombosporin and the matrix metalloproteases, they actually induces this synthesis of this new blood vessels through the angiogenesis. Okay. Here also this figure also in showing how the angiogenesis occurred, the formation of new blood vessel. Here these green ones are the tumor cells, these are the endothelial cells, and these are the supporting cells, these are the platelets, these red ones are the platelets, and this is the soluble protease basement membrane, and so on. Okay. Now let me go to the next problem. Problem number 14 says, which of the following is not an anthracycline anti-cancer agent? Your options are number A doxorubicin, number B epirubicin, number C darunavir, number D all of the above. Which of the following is not an anthracycline anti-cancer agent? Doxorubicin, epirubicin, darunavir, or all of the above? Which of the following is not an anthracycline anti-cancer agent? Which of the following is not an anthracycline anti-cancer drug? Doxorubicin, epirubicin, darunavir, or all of the above? Let me check the chat box. I got one answer saying number D. Let me check if number D is the correct answer or not. So the agent which is not an anthracycline anti-cancer agent is number C, darunavir. So darunavir is basically a antiviral agent. Okay, it is not a anti-cancer agent. It is an antiviral agent. So these are the anthracyclines, anti-cancer agents. Okay, these are the basic structure which has a planar ring system like this, and also it has a sugar moiety here. So as you can see, the anthracyclines, there is R1, R2, R3, and R4. Where R1, so these are the functional groups, and these are the doxorubicin, epirubicin, daunorubicin, and idarubicin. So these are the anthracyclines. So basically, intercalating agents, drugs contain a planar aromatic or heteroaromatic ring system which can slip into the double helix of DNA and distort its structure. Once bound, the drug can inhibit the enzymes involved in the replication and transcription processes. Examples include the dactinomycin and doxorubicin. Doxorubicin, previously called the adriamycin, belongs to a group of the naturally occurring antibiotics called anthracyclines. It was isolated from the Streptomyces pusitius in 1967, which is a similar structure to daunorubicin, different only in one hydroxyl group. However, this has an important effect on the activity, and the doxorubicin is one of the most effective anti-cancer agent was ever discovered. The drug intercalates into the DNA as an example of topoisomerase 2 in poison, as it stabilizes the complex formed between the DNA and topoisomerase 2, an enzyme that is crucial to the replication process. It is thought that an excessive number of these stabilized DNA enzyme complexes triggers the apoptosis because these enzymes are active during the cell growth and division. The topoisomerase 2 poisons are most effective against the rapidly proliferating cells. The second mechanism, which is doxorubicin, can prove harmful to DNA, involves the hydroxyquinone moiety, which can chelate iron to form an doxorubicin DNA iron complex. Reactive oxygen species are then generated, leading to single-strand breaks in the DNA chain. This mechanism is considered less important than the interaction with the topoisomerase 2, but it has been implicated that the cardiotoxicity of implicated in the cardiotoxicity of the doxorubicin. The third proposed mechanism involves the intercalated doxorubicin inhibiting helicases, which unravel the DNA into the single DNA strand. A variety of other anthracyclines are used in the cancer chemotherapy, mainly the daunorubicin is also called the cerubicin, and then the second generation anthracycline, epirubicin and idarubicin. Idarubicin lacks the methoxy group in R1, so as you can see here. So it's more polar and has the altered metabolism, which prolongs the half-life. The third generation anthracycline is being studied where the ammonium ion and of the sugar ring can be replaced with the azido group or the triazole ring. Such compounds do not appear to be susceptible to the efflux mechanism, which leads to the drug resistance, but also decreased general toxicity. Okay, so these are all about the intercalating agents. Now I will go to the next problem. Problem number 15 says, which of the following is not an third generation aromatase inhibitor? Your options are number A, anastrozole, number B, letrozole, number C, vorozole, and number D, fedrozole. Which of the following is not third generation aromatase inhibitor? Which of the following is not a third generation aromatase inhibitor? Anastrozole, letrozole, vorozole, or fedrozole? Let me know. A, B, C, or D. Which of the following is not a third generation aromatase inhibitor? Let me check the chat box. Okay, it's fine. Let me show you the correct answer and let us discuss it a little bit. The following, which is not a third generation aromatase inhibitor, will be number D, fedrozole. So aromatase inhibitors, they tend to be used as a second line drugs for the treatment of the estrogen dependent membrane bound complex consisting of a okay, breast cancer, a membrane-bound enzyme complex consisting of two proteins. One is the cytochrome P450 containing heme, and other is the reductase enzyme using NADPH as a cofactor. Aromatase cyclase catalyzes the last stage in the biosynthesis of estrogen from the androgens, where the aromatic ring is formed. The cytochrome enzyme contains heme, which serves to bind the steroid substrate and oxygen, then catalyze the oxidation. Since the enzyme catalyzes the last step of biosynthesis, it has been seen and it has been seen as an important target for the design of the anti-estrogenic drugs. Two types of inhibitor are used clinically: reversible competitive inhibitors and the irreversible inhibitors acting as a suicide substrate. Aminoglutamide is an early example of the reversible competitive inhibitor, has a disadvantages as it binds to the various cytochrome P450 enzymes and inhibits the range of the steroid hydroxylations. This results in undesirable side effects. Drug design based on the aminoglutamide is a lead compound resulted in more selective inhibitors such as the anastrozole, letrozole, which are used to treat breast cancer. The N4-nitrogen of the triazole ring interacts with the heme iron of the aromatase, prevents the binding of the steroid substrate. The anilio-nitrogen of the aminoglutamide serves as the same purpose. Formystine acts as a suicide substrate that permanently inactivates the aromatase and more selective in its action than the aminoglutamide. So these are the reaction catalyzed by the aromatase, the androstenedione and testosterone to the estrone and estradiol. And these are the reversible competitive inhibitor of the aromatase. These are the aminoglutamide. This is the anastrozole. This is the letrozole. So the first generation, so type 1 steroidal inhibitor, there is none. Type 2 are the aminoglutamide. The second generation is the formystine and the fedrozole and the rogatimate. And the third generation is the aromacin and the type 2 are the anastrozole, letrozole and borozole. So fedrozole, it is not a third generation, it is a second generation aromatase inhibitor. Okay. Now I will go to the next problem. Problem number 16 says kinase inhibitors must bind to the dash region of the enzyme. Your options are number A hinge, number B envelope, number C C-lobe and number D DFG region. Kinase inhibitors must bind to dash region of the enzyme. Hinge region, envelope, C-lobe or DFG region. Kinase inhibitors must bind to dash region of the enzyme. Hinge region, envelope, C-lobe, DFG region. So kinase inhibitors must bind to which region of the enzyme? If there is any answer in chat box, let me just check it once. I have one answer which is saying it number A. Okay, let me check the chat box now. Okay, sorry. So let's check the correct answer. So kinase inhibitors must bind to the very good hinge region of the enzyme. So kinases are basically the very comparatively new drug targets as the anti-cancer in the anti-cancer drug discovery. So what does this protein kinase, this enzymes do? They basically takes up one this phosphate from the ATP, converts it to ADP, and this phosphate it attacks in the protein substrate. So basically it phosphorylates the particular target protein. Okay, and the phosphorylation happens in a serine, tyrosine, or the threonine residue. So there are serine kinase, serine threonine kinase inhibitors or tyrosine kinase inhibitors are the major kinase inhibitors. Okay, and this is the phosphorylated drug targets. So this is the structure of the kinase enzyme. So basically we have a N-terminal lobe followed by a C-terminal lobe, and in between we have a particular region which connects this N-terminal lobe and C-terminal lobe, and this region is known as the hinge region. Okay, and also there is a DFG loop. Okay, and this DFG loop is present somewhere here. So these are the all regions. So any kinase inhibitor, so to be a to any molecule to be a potent kinase inhibitor, it must bind with the hydrogen bonding interaction with the hinge region. Why? Because this hinge region and this particular pocket is the ATP binding pocket, which is the substrate binding pocket. And when a kinase inhibitor it binds to this pocket, so the ATP now cannot bind, and the kinase enzyme cannot function. So basically, to be a kinase inhibitor, this particular so to be a basically the type 1 kinase inhibitor, to be specific, this particular inhibitors must bind to the hinge region of the enzyme. So there are several kinase inhibitors which are approved for the treatment. We have this is the air-based chart. You do not need to memorize it. I have just given it for your understanding. There are different several families of the kinase enzymes. So target families mainly are like TK, mTOR, RAF, VEGFR, MEK, ROCK kinase, PI3K, CDK, and antibody. And these are the nomenclature, how the naming happens. Okay, I'm not going to discuss it in detail, but it is a very important topic and very comparatively new anti-cancer drug therapeutics, the kinase inhibitors. Okay, so hinge was the correct answer. So now problem number 17, it says which of the following is not a PARP inhibitor? Your options are number A rucaparib, number B niraparib, number C lapatinib, number D olaparib. Which of the following is not a PARP inhibitor? Rucaparib, niraparib, lapatinib, and olaparib. Let me know A, B, C, or D. Which of the following you think is the correct answer? Which of the following is not a PARP inhibitor? Let me check the chat box. Okay, it's fine. I will just show you the answer and let us discuss it a little bit. So the following, which is not a PARP inhibitor, is number A. Okay, sorry, it will be number C. Lapatinib is not a kinase inhibitor. Sorry, is not a PARP inhibitor. Just let me just correct the numbering. Yeah. Okay. So number C is the correct answer. Lapatinib, because lapatinib is actually the kinase inhibitor. Okay. So what are the PARP? What is the PARP or what are the PARP inhibitors? So a number of research group are investigating the poly-ADP ribose polymerase inhibitors or PARP inhibitors as a potential anti-cancer agents. Although none have reached to the market to the date, the enzyme repairs single-strand breaks in the DNA, and so its inhibition can eventually result in the double-strand breaks in DNA and cell death. Some tumor cells are likely to be more susceptible to the PARP inhibitors than the normal cells. Okay, so these are the PARP inhibitors. So they have a shared benzamide core, which is like this one. Okay, and then differential R groups which conferring different size and flexibility. This is the olaparib, then rucaparib, then niraparib, and talazoparib. Okay, so these are the all the structures. Okay, one second. So these are the PARP targeted agents. So basically single target therapies like rucaparib, niraparib, talazoparib, pamiparib and fluzaparib, approved drugs, olaparib the first in class in the design. Okay, and then dual target therapies like PARP with the BCL2, BRD, HSP90. So all this is promising synergistic combination of the PARP with other anti-tumor agents, and the design is the pharmacophore of the PARP inhibitor and the pharmacophore of the other inhibitor. Modification gives the new tumor agents. So in recent years, PARP poly-ADP ribose polymerase have gained significant attention in the cancer treatment. Studies indicate that PARP play a crucial role in DNA repair, maintaining the genome stability, regulating cell activity, and responding to the DNA damage. Consequently, with the discovery and the exploration of PARP inhibitors have emerged in cancer treatment. These inhibitors are now used in clinical application for the tumor therapy, particularly for BRCA mutation-related cancers. A series of PARP inhibitors have been developed for the tumor treatment, with several approved for clinical use in cancer therapy. These inhibitors disrupt the DNA repair by inhibiting PARP enzyme activity, leading to cancer cell death, thus offering the potential for tumor therapy. We summarized the biological. So these are the other things about. So this is the about the poly-ADP ribose polymerase or PARP. Okay. Now problem number 18 says what sort of receptor is the muscarinic receptor? Your options are number A G protein coupled receptor, number B kinase linked receptor, number C intracellular receptor, number D ion channel. What sort of receptor is a muscarinic receptor? G protein coupled receptor, kinase linked receptor, intracellular receptor or ion channel receptor? I will wait for a minute only because we don't have much time left. So what sort of receptor is a muscarinic receptor? G protein coupled receptor, kinase linked receptor, intracellular receptor or ion channel? Let me check the chat box. I have got one answer which is saying it is number A. Let me check if number A is the correct answer or not. So, the muscarinic receptor is the number A G protein coupled receptor. So how do the G-coupled protein receptors work? So basically they are the largest class of the membrane proteins in the eukaryotes. They mediate most of the physiological responses of hormone, neurotransmitter and environmental stimulants. So when a G-protein coupled receptor is not bound to an agonist, it remains inactive. The G-protein also remains inactive in the cell membrane. Upon binding to a ligand or agonist such as the hormones or neurotransmitter, these undergo a conformational change activating the GEF domain. The GEF domain activates the monochromatic GP tails to replace the GDP from from a GTP in a GS alpha domain, activating the G protein. Upon activation, the GS alpha domain dissociates from the CRG protein complex and binds to the effector enzyme on the cell wall membrane to activate it. The activated effector enzyme generates the second messengers that activate the intercellular signal transaction cascades. The hydrolysis of the GTP into GDP in the GS alpha domain dissociates the GS alpha domain from the effector enzyme, deactivating the enzyme. So this is how the G protein coupled receptors work. And the muscarinic acetylcholine receptors. This is the types of the this the classification. The M1, M2, M3. Then the M1 is present in CNS salivary glands, the parietal cells, the M2 in heart, and M3 in the smooth muscle and exocrine gland. Okay. So the M4 and M5 are mainly present in the CNS, and the exact role is still unknown. So the M1 is give rises to the CNS excitation, memory, locomotor activity, gastric acid secretion. This M2 receptor they decreases the heart rate, force, and AV construction, and all smooth muscle contraction except the vasodilation and glandular secretion is increased by the M3. Okay. Now I will go to the next problem. Problem number 19 says anti-ulcer drug omeprazole is number A reversible proton pump inhibitor, number B antagonist of H plus K plus ATPase, agonist, number C agonist of H plus K plus ATPase, number D irreversible proton pump inhibitor. So what will be the correct answer? Anti-ulcer drug omeprazole is a which type of agent? Number A reversible proton pump inhibitor, number B antagonist of the H plus K plus ATPase, number C agonist of H plus K plus ATPase, and number D irreversible proton pump inhibitor. Omeprazole is which kind of agent? Let me check the chat box. Okay, let me show you the answer and let us discuss it a little bit. So, anti-ulcer drug omeprazole is number D, irreversible proton pump inhibitor. This is the structure of the omeprazole. So the proton pump is the only present in the canalicular membrane of the parietal cells and is crucial from the mechanism by which the hydrochloric acid is released into the stomach. It is called the H plus K plus ATPase because it pumps the protons out of the cell into the canaliculus. At the same time, it pumps the potassium ions back in. The energy required for this process is both proton and the potassium ions are being moved against their concentration gradients. In fact, the ratio of the protons inside the cell and the protons of the canaliculus is 1:10 to the power 6. The energy required to carry out this exchange obtained by the hydrolysis of the ATP, hence the term is ATPase. The pump is not responsible for the efflux of the chloride ion. This departs from the cell through the separate chloride ion channel. This outflow closely matches to the efflux of the protons, such as the chloride ion is released for every proton that is pumped out. As a result, hydrochloric acid is formed in the canaliculus rather than the inside of the parietal cell. As each chloride ion departs from the cell, the accompanied by a potassium ion which flows through this own ion channel. No energy is required for this outflow because the potassium ion is slowing down to the concentration gradient. The potassium ion acts as a counter ion for the chloride ion, and it's once it is in the canaliculus, it pumped back to the cell by the proton pump as I described previously. Consequently, potassium ions undergo a cyclic movement in and out the cell. So this is how this proton pump is secreting the role of proton pump is secreting the hydrochloric acid. Okay, so this is the lumen of the stomach, and these are the receptors, the H2M3 and CCK2. This is the proton pump, and these are the ion channels. Okay, and how the this is the canaliculus, and this is the lumen of the stomach. So this is the how the role of the proton pump in secreting the hydrochloric acid. So proton pump inhibitors, there are 4 PPIs in clinical use: omeprazole, lansoprazole, pantoprazole, and rabeprazole. The S enantiomer of omeprazole has also been approved recently. All PPIs have pyridyl methyl sulfonyl benzimidazole skeleton and as a prodrug, since they are activated in the rich acidic canaliculi to pair up the pyridyl cells. Once activated, they bind irreversibly to the exposed cysteine residues of the proton pump and block the pump, preventing the further release of the hydrochloric acid. There is a big strategic advantage in inhibiting the proton pump rather than blocking the histamine or cholinergic receptor. For example, H2 antagonists block the histamine receptors and they block the stimulatory effect of the histamine, but it does not block the receptors for acetylcholine or gastrin, and so it's still possible for the parietal cell to be activated throughout the secretion. The proton pump is downstream of all these targets and operates the final stage of hydrochloric acid release. Blocking it prevents the release of the hydrochloric acid regardless what mechanisms are involved in stimulating the hydrochloric acid secretion. Okay. So this was the problem number 19. So the now last question of today's session, it says, which of the following heterocyclic rings is present in cimatidine? Your options are number A imidazole, number B pyrazole, number C indole, and number D benzimidazole. Which of the following heterocyclic rings is present in cimatidine? Imidazole, pyrazole, indole, or benzimidazole? Which of the following heterocyclic rings is present in cimatidine? Let me know. Let me check the chat box. So I think I have one answer, number which is the B. Let me check if B is the correct answer or not. So the heterocyclic ring which is present in cimatidine is number A, imidazole. So these are the examples of the aromatic heterocyclic compounds. Okay, and remembering the names and structures are a little important in case of the medicinal chemistry. So these are the furan, pyrrole, thiophene, which are the 5-member ring with one heterocyclic atom. Pyridine, indole, benzofuran. We have carbazole, quinoline, isoquinoline. This is imidazole. So all the first ones are have only one heteroatom. Then imidazole, oxazole, pyrazole, pyrazine, pyrimidine, and these have the two heteroatoms, and purine has 4 heteroatoms. Okay, so the cimatidine inhibits the gastric acid release by acting as an antagonist to H2 receptors. It does not show any toxic side effects observed in the metamide and has been shown to be slightly more active. It has been found to inhibit the pentagastrin from stimulating release of the gastric acid. Pentagastrin is an analog of the gastrin, and the fact the cimatidine inhibits this suggests some relationship between the histamine and gastrin in the release of the gastric acid. Cimatidine was first marketed in the UK in 1976 under the trade name of the Tegamet, and it was the first really effective anti-ulcer drug doing away with the need of the surgery. For several years, it was the world's biggest selling prescription product until it pushed to the second place by ranitidine. So this is the how the synthesis of cimetidine happens. So the synthesis of cimetidine originally carried out by the four-step process where the lithium aluminium hydride was used as a reagent to initial reduction step. This one. Subsequent research revealed the reduction could be carried out more cheaply and safely using sodium and liquid ammonia, and so it became method to use for the manufacturing process. So this is first is this ester group is reduced to this particular your so sorry this carboxylate group is yes is reduced to the alcohol, and then we have this thiol, the amino aminothal, which gives rise to this, and then these are the steps they followed. Okay, so this is the imidazole ring which is present in the cimetidine. Okay, so this was the last question of today's session. So today majorly we discussed about the antiviral therapeutics, different antiviral drug targets, the mechanism of by which they work, and so on. Then we discussed about the anti-cancer and anti-ulcer drugs also. So it was the week 11 live session for this medicinal chemistry course. I will share my email ID here. Okay. So if you have any doubt or any query, please let me know through this email. Today it is our week 11 live session, and next Friday, we will have our last live session, which is our week 12 live session, followed by a summary session on 13th of April, which is on Sunday. So if you have any doubt, any query from this class or any previous class, please feel free to let me know through this email, because the last summary class will be on next week. So if you have any doubt or any query, I will try to prepare the slides accordingly. So please let me know if there is any doubts or query. Okay. So thank you for joining. Thank you for attending the class. I will close the session for today, and we will meet on next Friday from 4 p.m. In this live session. Okay. I am closing the session for today. Thank you.